Students, now we will discuss about biomolecules. Biomolecules are the chemical compounds which are found in living organisms. Now these chemical compounds which are found in living organisms, they include both organic compound and inorganic compounds. So first of all, we will discuss how to analyze the chemical compounds which are present in a living tissue. So here we have written some steps. Let's see what are the steps to analyze a chemical compound. First of all, we will take a living tissue. Now the living tissue, it could be a plant or an animal. So here we can take either a leaf or we can take a piece of liver. So the first step is to take a living tissue. Second step is that we have to grind that living tissue in trichloroacetic acid. So what we will do, we will transfer any living tissue into a mortar and pestle, and then we will add the tissue and trichloroacetic acid. After that we will grind it, clear? And after grinding it, we will get a thick slurry. So the third step is that we have to filter the slurry. When we will filter the slurry with the help of a cheesecloth, what will happen? We will get two parts. The part which is filtered down, it is known as filtrate, or it is also known as acid soluble pool. So the filtrate is acid soluble pool, and the substance which remains in that cheesecloth, it is known as retentate, or it is also known as acid insoluble pool. Clear? So the steps are, first of all, we will take the tissue, then we will grind it in mortar and pestle. We will take trichloroacetic acid in that mortar and pestle, and then we will grind the living tissue. We will get the thick slurry, and then we will filter it. The substance which will remain in the filter or which will remain in the cheesecloth, it is known as retentate, or it is also known as acid insoluble pool, and the substance which comes down is known as acid soluble pool or filtrate. Now second important thing is, what is filtrate? As I told you, filtrate is also known as acid soluble pool. It contains biomicromolecules. Biomicromolecules are those substances whose molecular weight is less than 800 Dalton. So all those compounds or substance which has molecular weight less than 800 Dalton, it will be the part of the filtrate. It includes amino acids, sugar, nucleotides, nucleosides. All these are biomicromolecules, and they will be the part of the acid soluble pool. Now the substance which remains in this cheesecloth, as I told you earlier, is known as retentate or acid insoluble pool. It contains biomacromolecules, or we can say they are polymeric substance. Clear? Now these biomacromolecules are those compounds whose molecular weight is more than 800 Daltons, and it includes protein, nucleic acid and polysaccharides. Clear? So we will get two parts. One is filtrate, or we can say acid soluble pool. In acid soluble pool, we have biomicromolecules which has molecular weight less than 800 Dalton, and it includes amino acid, sugar, nucleotide, or simply we can say it contains non-polymeric substance. In retentate, which is also known as acid insoluble pool, it contains those substances whose molecular weight is more than 800 Dalton. It includes protein, nucleic acids and polysaccharide. My dear student, one thing you have to remember always is that lipid is biomicromolecule, but it is found in the retentate which contains macromolecules. So lipid is not macromolecule, it is micromolecule, but it is found in the retentate because it forms aggregate. Clear? It forms aggregates. That's why it is found in the acid insoluble pool. So lipids are not macromolecules, but they can be obtained under macromolecule fraction due to their insoluble nature in aqueous medium of the cell. Clear? So this is an exceptional point which you have to remember. Now in your NCERT a table is given where they have given average composition of a cell. So just we will go through it. In a cell, water is 70 to 90% is water. Protein is 10 to 15%. Carbohydrate 3%. Nucleic acid 5 to 7%. Lipids 2% and ions 1%. So you have to just go through this table and you have to remember what is the average composition of the substance in a cell. So this was how to analyze the substance present in a living cell. Now we will study how to analyze the inorganic substances. So first of all, what we will do, we will take the fresh leaf. When we will take the fresh leaf, it is known as that it has the fresh weight. Clear? So we will take the leaf and it will have the fresh weight. Now this leaf contains organic substance, inorganic substance, and it contains water. Clear? The second step is that we have to evaporate all the water which is present in that leaf. So we will evaporate all the water which is present in that fresh leaf. Now we will have the dry weight of the leaf. Clear? In this dry weight we will have only the organic substance and inorganic substance. All the water is lost. After that, what we will do the combustion of the leaf, or simply we can say that we will do the oxidation. When the combustion will take place, what will happen? We will get the ash weight. Clear? We will get the ash which is known as ash weight. So what happened to this organic substance? This organic substance changes into CO2 and water. Clear? Now we will have only the ash, and this ash contains the inorganic substance. Now what are those inorganic substances which are present in the leaves? We can see it is given again in your NCERT in a table form. It contains sodium, potassium, magnesium, calcium, sulfate, phosphate. Clear? So if we have to analyze the inorganic substance present in a leaf, so what we will do? First we will take the leaf. This leaf contains all these three: organic substance, inorganic substance and water. The first thing is we have to remove all the water. So we have evaporated all the water. Now the dry weight of the leaf contains only organic and inorganic substance. When we will do the combustion of this leaf, what will happen? The organic substance will be changed into CO2 and water, and now we will have only the ash. Now we can analyze the ash and we will see that it contains all these substances. So these are the method of analysis. In my next video, we will study about the amino acids. Thank you.